Usually when we talk about a farm animal, there's this underlying expectation of simplicity, like, I don't know, a living lawnmower or something.
Right, yeah. You point a cow to pasture, it eats grass, and somehow out comes milk.
Exactly. It's very pastoral,
Right.
very comforting. We like things to be straightforward and natural and entirely uncomplicated.
Well, it feels very linear when you just look at it from the outside. Input grass, output dairy. I mean, it's a very safe, traditional mental model.
But when you actually step into the world of bovine neuroendocrinology and ruminal biochemistry, which we are doing today, that pastoral painting is just violently ripped away.
Oh, absolutely. It's completely shattered.
We're looking at a biological landscape that is, frankly, staggeringly complex.
Yeah.
It is the absolute definition of a hyper-optimized biological machine.
Yeah, calling it a machine is honestly the best way to frame it.
So today, for you listening, we are setting out to completely shatter that notion of a cow as a simple grazing animal. We're going to deconstruct this organism from the outside in.
And we are not skipping the dense science either.
No, not at all. We are diving into the deep end. Because by the time we're done, you're going to see this animal not as livestock, but as this finely tuned bioreactor. I mean, a system that actively manipulates time, light, and microbiology just to survive and produce.
It really is a feat of evolutionary and agricultural engineering. But to understand how those internal systems operate, you first have to understand the specific data the animal is actually receiving from its environment.
Right, the inputs.
Exactly. You have to look at the bovine sensorium. Because cows process the physical world drastically differently than humans do.
I mean, their eyes are practically on the size of their head.
Yeah, and vision is actually their dominant sense. They obtain almost half of their information visually, but it is an entirely alien visual experience compared to ours.
Yeah, I was looking at the anatomical structure of their eyes in the sources, and that lateral placement on the skull gives them this massive panoramic field of view. It's about 330 degrees, right?
Yep, roughly 330 degrees.
Which is wild, because if you're a human, your field of view is roughly 140 degrees. So a cow can essentially see almost everything around it without ever turning its head.
Which makes perfect sense as an evolutionary anti-predator adaptation. I mean, if you're a prey animal on an open plain, you really need to know if a wolf is sneaking up behind you.
Right. But there's a huge trade-off there, isn't there?
There is a massive trade-off. They sacrifice stereopsis for vigilance.
Stereopsis being depth perception.
Correct. Because their eyes are situated so far apart on the sides of their head, they have a very, very narrow binocular overlap.
Binocular vision is where both eyes look at the same thing at the same time, right?
Yes, and that is the mechanical requirement for the brain to triangulate distance and actually perceive depth. For humans, that overlap is huge. But for a cow, it's only about 25 to 50 degrees directly in front of their nose.
Wow.
So for the vast majority of that sweeping 330-degree visual field, they're seeing the world entirely monocularly, flat.
So it's essentially like having a highly advanced panoramic camera. But the autofocus is just broken for anything outside of the dead center.
That's a highly accurate way to visualize it, yeah.
I mean, it's fantastic for spotting a predator moving on the horizon at 200 yards. But it's absolutely terrible for judging how deep a puddle is right in front of your own hooves.
Exactly. And the physical apparatus of the eye just compounds that issue. They have these distinctive horizontal oval pupils.
Which looks super weird close up.
They do, but they're great for scanning a wide horizon. The problem is the ciliary muscles, the muscles that actually squeeze and pull the lens of the eye to change its focus, those are relatively weak in a bovine.
So they can't change focus quickly.
Right. They are incredibly slow at accommodation. So if they shift their gaze from a distant mountain to like a gate right in front of them, it takes significant biological time for the lens to physically reshape and bring that gate into focus.
Okay, so their depth perception is awful, their focus is slow. What about color? Because we constantly see those classic images of the matador waving the bright red cape at the bull. They must see red, right?
They actually don't. They don't see it at all. They are dichromatic.
Wait, really? So the red cape is a total myth?
Total myth. Like most mammals, but unlike humans who have trichromatic vision, cows possess only two distinct classes of cone photoreceptors in their retinas.
So what colors can they actually process?
Well, their peak spectral sensitivities hit around 450 nanometers, which is the blue-violet range, and 550 nanometers, which is the yellow-green range.
Let me pause and make sure I'm visualizing this right. Because they lack the specific cones required to process the longer wavelengths of light, the red end of the spectrum, What happens when they look at, say, a bright red barn or a green tractor?
The visual cortex simply doesn't have the hardware to differentiate those specific wavelengths.
It just looks muddy.
Yeah, reds and greens essentially blur together into muted shades of brown, yellow, or gray. So, going back to your example, the matador's cape isn't enraging the bull because it's red.
It's just the movement.
Exactly. The bull is reacting to the aggressive, high-contrast movement of the fabric. But, what they lack in vivid color distinction, they absolutely make up for in contrast sensitivity and low-light vision.
Right, that's where the tapetum lucidum comes in.
Yes.
It's that reflective layer behind the retina. Like we've all been driving at night and seen a cat's or a cow's eyes practically glowing in the headlights.
That glow is the tapetum lucidum doing its job. It acts like a biological mirror.
Because normally, light just hits the back of the eye and gets absorbed, right?
Right. When photons of light enter the eye and pass through the retina, some of them inevitably miss the photoreceptors.
Yeah.
In a human eye, those photons are just absorbed by the dark tissue and lost. But in a bovine eye, they hit that reflective layer and bounce back through the retina a second time.
Giving the photoreceptors another chance to capture them.
Exactly. It dramatically amplifies their night vision. But here's the catch. During the bright light of day, this exact same mechanism creates a severe sensitivity to high contrast environments.
And this is where the physical reality of their vision gets truly wild for me. When you combine that high contrast sensitivity, the incredibly poor depth resolution, and a slow focusing mechanism, a simple shadow stops being just a shadow.
To them, it becomes a physical barrier.
That is so strange to think about.
It is. To the bovine visual cortex, a sharp line on the ground between bright sunlight and a dark, crisp shadow cast by a roofline, it doesn't register as a trick of the light.
Because they lack the depth perception to see that the ground continues flat underneath it.
Right, so that stark black line registers as a physical, bottomless pit, a sheer drop-off.
Which explains so much about herd behavior. I mean, if their vision is that limiting for depth and shadows literally look like cliffs, how do they navigate uneven terrain without constantly panicking or falling over?
They rely heavily on behavioral compensation. If you watch a cow investigating something unfamiliar or walking on rough ground, you'll notice they immediately lower their heads.
Oh, to use that tiny window of depth perception.
Precisely. By dropping their skull, they are deliberately aiming that tiny 25 to 50 degree window of binocular depth perceiving vision directly at the ground in front of their feet.
They physically sweep their only depth perceiving tool across the obstacle.
Yes, and this is a fundamental principle of safe livestock handling. You have to minimize high contrast shadows in their walking paths, and you absolutely must avoid surprising them in the massive blind spots directly behind them.
Because they'll just panic.
Exactly.
Okay, so vision is just one piece of this sensory puzzle. Let's talk about their olfaction, their sense of smell, because according to the sources, it's working in absolute overdrive. They use their noses not just to find food, but as this complex chemical communication network.
Oh, their olfactory system is deeply tied into their social and survival circuitry.
They can actually detect alarm chemicals in the urine of stressed herd mates, right?
Yes. They can identify conspecifics other cows entirely by smell, and they remember individuals for years. But the detection of stress pheromones is a critical survival mechanism.
How does that work chemically?
When an animal is stressed by pain, fear, or illness, its body alters its metabolic exhaust. It releases specific volatile chemical markers in its urine and feces.
So they literally sweat out their fear.
Essentially, yes. And other cows detect this, which instantly elevates their own physiological reactivity to potential threats. They process these complex chemicals using a very specific physical maneuver called the Fleeman response.
The lip curl. You see them stretch their neck out and roll their upper lip way back.
That's the one. And that curl serves a highly specific mechanical purpose. It physically seals off the nasal passages and creates a negative pressure vacuum
To suck the air in.
to trap those heavy pheromone molecules and actively pump them up into the vomeronasal organ.
The vomeronasal organ, where is that exactly?
It's a specialized tubular chemoreceptor located just above the roof of the mouth, right at the base of the nasal cavity, and it's wired directly into the amygdala and hypothalamus in the brain.
So it bypasses the normal smelling centers.
Completely. It allows them to literally taste the emotional and chemical state of their entire herd on a subconscious level.
Wow. So their entire sensory apparatus, the sweeping panoramic monocular vision, the intense sensitivity to contrast, the chemical sampling of the herd's stress levels, it's all this perfectly evolved array designed to keep them alive and vigilant so they can safely do one thing: ingest food.
That is the ultimate goal.
But what happens once that food actually travels down the esophagus?
Yeah.
That is where the true biochemical marvel of this animal begins. We're moving from the sensory array to the engine room, the rumen bioreactor.
And calling it a bioreactor is no exaggeration at all.
It's basically a massive, 40-gallon vat.
Yes, a 40-gallon, constantly churning, strictly anaerobic fermentation vat. And the fundamental irony of the cow is that when we talk about a cow eating grass, the cow isn't actually digesting the structural carbohydrates in that grass.
Wait, they don't digest the grass?
No. The bovine genome itself completely lacks the enzymes required to break down cellulose and hemicellulose.
That is wild. So the cow is essentially just a giant walking life support system for a microbiome. It just feeds this highly complex microbial consortium of bacteria, protozoa, fungi, and archaea.
That's exactly what it is.
Okay, let's get down to the physical biochemistry of this because it is intense. The cow ingests these complex dietary carbohydrates, the cellulose, the hemicellulose, the starches. What exactly are the microbes doing to that plant matter in the stomach?
The microbes deploy an arsenal of enzymes, specifically glycoside hydrolases, to physically shear those complex, tightly bound polysaccharide chains down into their constituent simple sugars.
Meaning hexoses and pentoses.
Exactly. 6 carbon and 5 carbon sugars. Once they have these basic building blocks, the microbes absorb them. Inside the microbial cells, the pentoses are converted to hexose and triose phosphates via the transcatallase and transaldolase reactions.
Okay, let's ground those terms for a second. Transcatallase and transaldolase. These are essentially enzymes acting like molecular mechanics, right? They're just shuffling carbon atoms around to turn 5 carbon sugars into 6 carbon sugars so the cell can actually process them.
That's exactly what they're doing. They are standardizing the fuel so it fits into the next pathway.
Which is?
Well, from there, the bulk of these intermediates are metabolized into pyruvate, and this occurs almost exclusively via the Embden-Meyerhof pathway.
Which is just the classic sequence of glycolysis.
Yes, standard glycolysis.
But physically, what's happening to that sugar molecule in this dark, oxygen-free vat? Because, in our bodies, humans use oxygen to burn that sugar down to nothing but water and carbon dioxide.
That is the crucial difference. Because the rumen is a strict anaerobic environment, meaning absolutely zero oxygen, the pyruvate cannot enter a traditional Krebs cycle.
Because there's no oxygen to catch the electrons at the end.
Right. The microbes can't fully burn the fuel because they don't have oxygen to act as the final electron acceptor at the end of the chain. So instead, they are forced to undergo fermentation.
You just sort of tear it apart halfway.
Basically, they tear the sugar apart just enough to extract a tiny bit of ATP for themselves, and then they dump the partially broken down carbon molecules back out into the rumen fluid. And the end products of this fermentation are volatile fatty acids, or VFAs.
Ah, the VFAs. The big 3 being acetate, propionate, and butyrate.
Those are the big 3, yes.
And these VFAs, which are essentially microbial waste products, they are the actual energy currency of the cow. The cow absorbs them, and they provide up to, 70% of the host animal's total energy requirement?
Up to 70%, yes.
It seems elegant enough. The microbes eat the sugar, they excrete VFAs, and the cow absorbs the VFAs. But the balance of which specific VFAs are produced is practically a matter of life and death, isn't it? Because this all comes down to the underlying hydrogen economy of the rumen.
Yes, we have to view the rumen through the lens of thermodynamics. Specifically, it's governed by hydrogen partial pressure.
Okay, walk me through that.
When the microbes ferment fibrous plant matter, like grass, into acetate or butyrate, those specific biochemical pathways are net generators of metabolic hydrogen.
That make extra hydrogen.
Right. They physically strip hydrogen atoms off the carbon skeletons and release free H2 gas directly into the rumen fluid.
And if too much free hydrogen builds up, the whole fermentation vat just shuts down.
It acts as a thermodynamic roadblock.
How does a gas act as a roadblock?
Well, if H2 accumulates in the fluid, it exerts back pressure on the chemical reactions. It physically hinders the reoxidation processes of critical coenzymes like NADH and FADH2.
Ah, the electron carriers.
Exactly. If the microbes can't regenerate those coenzymes, they can't perform glycolysis. Microbial metabolism simply halts.
The battery can't recharge.
So, to prevent this catastrophic buildup, the rumen relies on a very ancient class of microorganisms, the methanogenic archaea.
The methanogens.
Yes. These methanogens act as a vital hydrogen sink. They aggressively consume that excess free H2 and use it to reduce carbon dioxide, producing methane, CH4.
And then the cow just burps it out.
Yes, the cow eructates or burps out this methane to clear it from the system entirely.
Okay, so methane isn't just some random, unfortunate byproduct of digestion that we want to get rid of for climate reasons.
Hmm.
It is a critical, life-saving exhaust valve for the excess hydrogen generated by making acetate.
Absolutely vital.
But there is another pathway, right? A metabolic route that actually consumes hydrogen instead of generating it.
Yes, and that is the production of propionate.
Okay, how is that different from acetate?
When the rumen microbes ferment rapidly degradable carbohydrates, think starches from corn or grain, they synthesize propionate. And they do this via two main routes, the succinate pathway or the acrylate pathway.
Succinate and acrylate.
Got it. The brilliant part of both of these metabolic routes is that they physically incorporate free hydrogen into the final propionate molecule.
So they use the hydrogen up.
Exactly. Therefore, propionate production acts as an alternative hydrogen sink. It competes directly with the methanogens for the available H2 in the vat.
Which brings us to an absolute fascinating tangent in the research we reviewed. There was this dense, highly detailed in vitro study that attempted to hack this exact hydrogen dynamic. They wanted to shut down the methane without killing the cow's energy supply. Walk me through what these researchers actually did with 2-bromothane sulfonate.
Right, the BES study. It's a remarkable piece of experimental biochemistry. The researchers wanted to directly inhibit those methanogenic archaea to stop greenhouse gas emissions.
Which makes sense, climate change and all.
So they introduced a chemical analog called 2-bromothane sulfonate, or BES, directly into the simulated rumen fluid.
What does BES do?
BES is highly specific. It binds to and completely blocks the enzyme systems that are unique to methanogens. And on the surface, it worked phenomenally well. It slashed methane production by nearly 85%.
But wait, if you just established that the methanogens are the exhaust valve for the hydrogen, and you plug the exhaust pipe with this BES chemical, doesn't the hydrogen have nowhere to go?
That is exactly the fundamental thermodynamic trap they ran into.
The system's stalled.
Entirely. By chemically paralyzing the methanogens, the metabolic hydrogen rapidly accumulated. The fibrolytic bacteria, the ones digesting the fiber, they sensed this massive spike in H2 partial pressure, and just as we discussed, it suppressed their normal fermentation pathways.
So they stopped eating the grass.
Yes, the total production of volatile fatty acids absolutely plummeted. So yes, you successfully stopped the methane, but you also completely starved the cow of its primary energy source. The biological engine stalled out.
So to fix this, you can't just plug the exhaust pipe. You have to essentially hire new workers to use up that accumulating hydrogen. You have to redirect the flow of the factory to a different product.
And that product had to be propionate.
Right, because making propionate consumes hydrogen.
Exactly. So the researchers' brilliant solution was to introduce a highly concentrated, lab-grown consortium of propionate-producing bacteria simultaneously with the BES inhibitor.
They just dumped new bacteria in.
They utilized 4 very specific strains to hijack the hydrogen. They used Lactoplanta Bacillus plantarum and Megasphaera elsdeni to drastically reinforce the acrylate pathway.
Okay, those are 2 of them.
And they added Selenomonas ruminantium and Acidopropianobacterium thenia to flood the succinate pathway. They seeded this consortium into the fluid at a massive concentration. We're talking 10 billion cells per milliliter.
10 billion per milliliter. That is a staggering microbial payload. So what actually happened? Did the new workers take the job?
The results were profound. When they combined the chemical inhibitor with this engineered bacterial consortium, methane emissions dropped by up to 94% compared to the control group.
94%.
Yes. But the crucial victory was that the total VFA profiles were entirely restored.
The energy came back.
The introduced bacteria successfully captured the excess metabolic hydrogen and physically forced it into the synthesis of propionate.
So they captured the exact same hydrogen that the archaea used to turn into useless methane gas, and they turned it into a high-energy fat that the cow can actually absorb and use to make milk or muscle. That is biological hacking at its absolute finest.
It really is. Furthermore, they used advanced sequencing to track the underlying ecological shift in the rumen microbiome.
What did they find?
Well, while the overall richness of species, meaning how many different types of bugs were present, remained stable, the dominance of certain groups completely inverted.
The balance of power shifted.
Drastically. They saw a massive decrease in typical fibrolytic bacteria and a massive, statistically significant surge in the relative abundance of a very specific endogenous bug called Succinoclasticum.
Succinoclasticum, I love that name. It literally implies it specializes in smashing succinate to turn it into propionate.
Exactly. The addition of the probiotic consortium acted as a catalyst that modulated the cow's own endogenous microbiome. It permanently enhanced the entire propinet metabolic network so it could effortlessly handle the massive hydrogen load left over by the BES.
Okay, so through all this intense fermentation, the vat is churning out these volatile fatty acids, acetate, propionate, butyrate. Now, how does the cow actually get them out of this dense 40-gallon fluid matrix and into its bloodstream? Because this isn't just passive soaking, right? The rumen wall itself isn't just a leather bag. It is highly metabolically active tissue.
Oh, very active. The transepithelial transport of these acids is a highly coordinated, multi-layered process.
Let's break down the wall.
The rumen is lined with a stratified squamous epithelium. It consists of 4 distinct cellular layers. You have the stratum corneum on the very surface, followed by the stratum granulosum, the stratum spinosum, and resting on the stratum basal.
So 4 layers of cells.
Yes, and every single molecule of VFA has to navigate this living barrier to reach the portal vein.
And they have 2 primary methods for crossing this barrier, correct?
They do. First, there is the simple passive diffusion of the undissociated lipophilic form of the VFAs.
Undissociated meaning they don't have an electrical charge.
Exactly. Because they are technically fats, if they aren't ionized, they can slip right through the lipid bilayers of the cell membranes without any help.
But the fluid in the stomach is acidic, isn't it?
Right. The pH of the rumen fluid typically favors the ionized anionic form of these acids.
Meaning they carry a negative charge.
Yes, and a charged molecule will bounce right off a lipid cell membrane. So to get those across, the epithelium utilizes extensive carrier-mediated transport.
Protein pumps.
It deploys transport proteins like NHE3.
What's NHE3 do?
It's a sodium-hydrogen exchanger. It physically swaps a sodium ion for a proton to help buffer the local pH. And it works alongside MCT-D and MCT4.
And what are those?
Those are specialized monocarboxylate transporters. They actively shuttle the charged VFA molecules deep into the tissue.
But here's the part of the literature that truly stopped me in my tracks. The rumen wall doesn't just passively act as a doorway for these acids. It actively eats them as they pass through.
Yes, it does.
Specifically, it eats the butyrate.
This introduces a critical physiological concept known as metabolic pull. As the butyrate passes through the deeper layers of the tissue, the stratum spinosum and stratum basal, the epithelial cells heavily metabolize it.
They just burn it for fuel.
Yes. Transcriptomic analyses, looking at the actual genetic expression of the rumen epithelium, show massively upregulated expression of specific metabolic genes. They rely heavily on BDH1.
BDH1.
It's an enzyme that encodes 3-hydroxybutyrate dehydrogenase, and they also rely on HMGCS2.
I recognize HMGCS2. That's the master control gene for ketogenesis in the liver.
It is. The rumen epithelium preferentially burns butyrate for its own local cellular energy needs, essentially powering the transport pumps we just talked about. But it takes the remainder of the butyrate and physically converts it into ketone bodies right there inside the stomach wall.
Wow.
Specifically beta-hydroxybutyrate, or BHB. It then releases this BHB into the portal blood.
Why go through all that trouble? Why not just let the butyrate pass through?
This intense local metabolism is vital. By instantly destroying the butyrate as soon as it enters the tissue, the cells maintain a steep concentration gradient.
Oh, it creates a vacuum.
A chemical vacuum, exactly.
Wow.
It continuously pulls more and more butyrate out of the rumen fluid. This ensures rapid absorption and prevents the acids from building up and crashing the entire ruminal pH.
It's such an incredibly elegant, self-sustaining system. I mean, the microbes make the highly acidic fuel, and the stomach wall burns a little bit of that exact fuel just to power the biological pumps that vacuum the rest of it in so the stomach doesn't become too acidic. It's genius.
It is a perfectly tuned loop.
Okay, so we've thoroughly covered the carbon and the hydrogen cycles. Now we have to look at the nitrogen. Because to keep this massive 40-gallon microbial workforce alive, reproducing and digesting grass, the cow has to masterfully manage nitrogen to build proteins.
Which leads us to urea nitrogen salvaging.
Right. And if we step back and look at normal mammalian physiology, humans, dogs, pigs, the breakdown of dietary proteins and cellular amino acids generates ammonia as a byproduct.
And ammonia is highly neurotoxic.
Extremely.
To survive, the mammalian liver rapidly extracts this ammonia from the blood and converts it into urea, which is a water-soluble, relatively inert, and non-toxic compound.
And in almost all non-ruminants, this urea is viewed strictly as a final waste product. It travels to the kidneys and is excreted entirely in the urine. Flush it away.
But the bovine looks at that exact same urea and says, why would I throw away perfectly good fertilizer?
Exactly.
It is arguably the most efficient nitrogen recycling program on the planet. Depending on the protein content of their diet, bovines can salvage and recycle an astonishing 29% to 99% of their hepatic urea nitrogen directly back into their gastrointestinal tract.
99%. Bypassing the kidneys almost entirely.
Entirely.
Let's track the physical biochemistry of that journey. Ammonia, generated down in the rumen and from systemic muscle breakdown, hits the liver via the bloodstream. How does the liver process it so safely without letting it reach the brain?
Within the microanatomy of the hepatic lobule, the liver utilizes a brilliant two-stage spatial arrangement. It ensures zero ammonia escapes into the general circulation.
What's the third stage?
The periportal hepatocytes. These are the liver cells situated right at the entry point of the blood supply. They capture the vast bulk of the incoming ammonia.
And what do they do with it?
They process it through the incredibly energy-intensive ornithine cycle to synthesize urea.
But biological systems aren't 100% efficient. What about the ammonia molecules that slip past those first periportal cells?
That is where the second stage comes in. The escaped ammonia is mopped up by the perivinous hepatocytes.
And where are those?
They're located at the very exit of the liver sinus, right before the blood heads to the heart. And these specific cells express incredibly high levels of an enzyme called glutamine synthetase.
So they act like a filter of last resort.
They act as a chemical sponge, capturing any residual toxic ammonia and instantly converting it into the safe amino acid glutamine.
Okay, so the liver has efficiently and safely packaged the nitrogen into urea. Normally this goes to the bladder. How does the cow physically get it back into the rumen to feed the bacterial workforce?
It relies on specialized transport channels, facilitative urea transporters known as UTA and UTB.
Where are those located?
In a cow, these transporter proteins are densely, uniquely expressed in two critical locations: the deep epithelial layers of the rumen wall and the salivary glands in the mouth.
The salivary glands.
Yes. These transporters allow the high concentration of urea in the bloodstream to diffuse straight through the tissues. The cow actively secretes massive quantities of urea directly into its saliva.
So a cow is literally constantly swallowing its own filtered urea,
Yes.
while simultaneously pumping it directly out of its bloodstream, through its stomach lining, and into the fermentation vat.
Yeah.
That is insane.
But highly efficient.
But wait, the microbes themselves can't actually build proteins out of urea directly, can they? They need raw ammonia.
That's the missing link, and this is where a highly specialized symbiotic subpopulation of microbes comes into play. The epithelial wall of the rumen isn't just smooth tissue. It is heavily colonized by a dense biofilm of urease-producing bacteria.
Bacteria that specifically make urease.
Yes. These bacteria literally hug the stomach wall. As soon as the urea molecules cross the stratum corneum from the bloodstream, these wall-hugging bacteria deploy the urease enzyme to violently hydrolyze the urea back into ammonia, NH3, and carbon dioxide.
So it's a localized delivery system. The stomach wall secretes the safe urea. The specialized bacteria living right on that wall instantly smash it back into raw, usable ammonia.
And then the general population of microbes swimming in the vat seizes that free ammonia to start building amino acids.
It's unbelievable.
The general microbial population uses that recycled nitrogen to synthesize microbial crude protein. They use it to multiply and build their own cellular bodies.
And then what happens to them?
Eventually, the fluid dynamics of the stomach wash these billions of microbial cells out of the rumen, through the subsequent stomach compartments, and down into the small intestine.
Where the cow digests them.
Exactly. There, the cow finally deploys its own mammalian gastric juices to digest the bacteria, absorbing incredibly high-quality essential amino acids.
I have to admit, it's just amazing that a cow effectively eats its own internal waste products by feeding it to an internal farm of bacteria and then just eats the bacteria.
It is nature's perfect loop.
But surely there has to be a physical catch. I mean, is there a metabolic cost to running this constant detoxification and recycling loop? You're running the ornithine cycle in the liver nonstop?
Oh, there is a severe, strict thermodynamic cost. The synthesis of urea via the ornithine cycle requires massive amounts of ATP. It burns cellular energy.
So it's expensive.
Very. If a ruminant is out on a pasture eating a high forage diet that is very high in rapidly degradable protein, the rumen microbes will rip that protein apart and produce excess ammonia incredibly fast.
And the liver has to handle all that.
The liver then has to expend immense amounts of energy to convert that toxic ammonia surge into urea.
But if the diet is just leafy forage, there isn't enough dense energy like starch from corn in the stomach for the microbes to actually use all that recycled urea, right?
Exactly. The microbes lack the fermentable carbohydrate energy required to capture that nitrogen and build protein.
So what happens?
So the liver burns its own energy reserves to synthesize urea, pumps it into the rumen, the microbes can't use it, it turns back to ammonia, it goes back to the liver, and the cycle repeats until the urea is finally excreted in the urine.
That sounds exhausting.
It's a futile energy-draining cycle. This explains why forage-fed cattle can sometimes exhibit poor overall nitrogen retention and lose weight compared to cattle-fed precisely balanced, high-concentrate diets. It perfectly highlights the delicate, precarious balance of ruminant nutrition.
And this precarious balance is the perfect transition to the darker side of this biology, because this incredible, elegant efficiency works flawlessly in a natural grazing environment, producing just enough milk for a single calf.
Right.
But when we step into modern dairy production, we ask this biological machine to produce massive, staggering amounts of milk. And that singular demand pushes this metabolic engine past its breaking point, straight into the realm of ketosis and hypocalcemia.
To truly understand the pathology of ketosis, we have to look closely at the immediate periprurient period.
The transition period.
Yes, the transition period spanning the final weeks of pregnancy and the first few weeks after calving. A modern high-yielding dairy cow has been genetically selected to prioritize milk synthesis above her own bodily preservation.
She will literally milk herself to death.
Yes. Upon calving, she will suddenly ramp up milk production to an extent that physically exceeds her gastrointestinal capacity to consume enough dry matter to support it.
She can't eat enough.
The energy demand curve goes almost vertical, while the physical feed intake curve severely lags behind.
This creates a state of negative energy balance, or NEB.
Yeah.
I always try to visualize NEB like trying to run a smartphone with the screen on maximum brightness, streaming a high-definition video over cellular data while running a massive 3D game in the background.
That's a great analogy.
The battery inside the phone is draining vastly faster than the tiny charger plugged into the wall can possibly refill it.
The math simply doesn't work in the cow's favor, and when her hypothalamus and endocrine system realize that the dietary energy entering the rumen is critically insufficient, she triggers a systemic emergency protocol.
Which is?
The massive, rapid mobilization of her own body fat. The adipose tissue begins to break down stored triglycerides, releasing them into the bloodstream in the form of non-esterified fatty acids, or nephas.
Let's dig into the molecular triggers of that fat breakdown, because the sources we reviewed isolated a very specific transcription factor controlling this whole cascade, TFB.
Yes, transcription factor EB. In cows experiencing the onset of ketosis, molecular biologists found a marked, alarming translocation of TFB.
Meaning it moves.
Right. Normally, it resides quietly in the cytoplasm of the fat cells. But during this severe energy crisis, it physically moves into the nucleus of the adipocytes.
And what does it do once it's in the nucleus?
Once inside the nucleus, TFE acts as a master genetic regulator. It binds to the DNA and massively upregulates the transcription of multiple lipase genes.
Lipases being the fat-cutting enzymes.
Exactly. These newly synthesized lipases act like biochemical scissors, actively accelerating the shearing of stored fat, completely flooding the vascular system with nephas.
And all of those circulating nephas head straight to the liver to be burned for emergency energy. But the liver, despite being this incredible metabolic hub, physically cannot handle the volume, can it?
The hepatic oxidative capacity becomes entirely overwhelmed. The liver mitochondria desperately try to perform beta oxidation on these incoming fatty acids to generate ATP.
But there's a problem.
There's a huge problem. And here is where the rumen biochemistry comes back to haunt the cow. The Krebs cycle inside the liver mitochondria requires oxaloacetate to function.
Oxaloacetate, okay.
It's a molecule derived from glucose. Because the cow is eating very little, the rumen isn't producing enough propionate.
Right, because propionate is the hydrogen sink from grain.
Exactly. And without propionate, the liver can't perform gluconeogenesis to make glucose, meaning it lacks oxaloacetate.
So the whole cycle stops.
The Krebs cycle physically bottlenecks. The fat is broken down into acetyl-CoA, but that acetyl-CoA has nowhere to go.
It's a traffic jam at the molecular level. So to clear the backlog and stop the liver from shutting down, it converts all that stalled acetyl-CoA into ketone bodies.
Primarily beta-hydroxybutyrate, BHB, acetoacetate, and acetone. The liver exports these ketones into the blood, hoping the brain and muscles can burn them.
But they can't burn them fast enough.
Exactly. When the sheer production of these ketone bodies vastly exceeds the peripheral tissue's ability to utilize them, they accumulate. The blood becomes thick with them. That is the definition of hyperketonemia or clinical ketosis.
And reading through the veterinary metabolomics, it became clear there isn't just one monolithic type of ketosis. There's type I and type 2. What's the physiological difference?
It fundamentally comes down to timing, liver architecture, and insulin dynamics. Type I is what we call classic nutritional ketosis.
What does that hit?
It typically hits 3 to 6 weeks postpartum, right when the physical volume of milk yield reaches its absolute peak. The primary driver is an absolute, unavoidable caloric deficit.
Starvation, basically.
Yes. The cow experiences severe hypoglycemia, dangerously low blood sugar, because her liver simply cannot manufacture enough glucose to keep pace with the udder.
So type 1 is essentially acute starvation in the face of massive output. What about type 2?
Type 2 is far more insidious, much harder to treat, and usually strikes much earlier, often within the first week postpartum or even right before calving.
Why does it happen so early?
It is fundamentally linked to fatty liver disease. In these cows, usually ones that were slightly overweight before calving, the liver takes up so many mobilized nephas that it physically cannot oxidize them fast enough.
And it can't export them either.
No, it can't package them into very low-density lipoproteins, VLDLs, to export them safely. So the fat literally pools and crystallizes inside the hepatocytes.
Ouch.
It physically distends and damages the liver's microscopic architecture.
And if you pull blood from a type 2 cow, the profile looks completely different from the starving type of cow, right?
Yeah.
Completely different. Instead of severe hypoglycemia, cows with type 2 ketosis often exhibit hyperinsulinemia and deep systemic insulin resistance.
Way high insulin.
Yes. They might actually test with normal or even elevated blood glucose levels, but their peripheral tissues stubbornly refuse to respond to the insulin signal. It mirrors human type 2 diabetes.
That is fascinating.
It's a profound, complex metabolic dysfunction rather than just a simple math problem of calories in versus calories out.
I think we need to establish the exact clinical thresholds for this, because this isn't a binary the cow is sick or the cow is healthy situation. There's a sliding scale of danger.
Oh, definitely. Subclinical ketosis, which is terrifying because it can silently affect up to 40% of a dairy herd and severely depress their immune systems without any obvious visual signs.
The silent killer.
Right. It is generally defined as blood BHB levels greater than or equal to 1.2 millimoles per liter.
And what about clinical?
Clinical ketosis, where the cow crashes, she stops eating, rapidly loses body condition, her milk yield plummets, and you can literally smell the sweet chemical odor of acetone on her breath that typically presents with BHB levels spiking at or above 3.0 millimoles per liter.
But the ketones themselves, the BHB, aren't just passive markers of starvation. The research highlighted that high levels of nephas and BHB are actually active, toxic signaling molecules. They do physical damage.
That is a vital point that shifts our entire understanding of the disease. High concentrations of nephas and BHB act as metabolic danger signals to the immune system. They directly trigger systemic inflammation by physically activating a protein complex called NF-kappa B.
Let's define NF-kappa B. Why does that protein complex matter? Does the body physically mistake these fat molecules for a bacterial infection?
In a way, yes. NF-kappa B is the master transcriptional switch for the inflammatory response. When the high fat levels flip that switch, the immune cells begin expressing potent pro-inflammatory cytokines.
Like what?
Specifically tumor necrosis, factor alpha, TNF-alpha, and interleukins like IL-6. The body essentially declares war on itself, establishing a state of chronic, low-grade systemic inflammation.
Which just makes everything worse.
It actively worsens the insulin resistance, damages tissue, and exhausts the white blood cells, which is exactly why a ketotic cow is massively, disproportionately susceptible to severe bacterial infections like mastitis in the udder or metritis in the uterus.
And as if that cascade wasn't devastating enough, it gets exponentially worse when you introduce the primary comorbidity, hypocalcemia, otherwise known as milk fever.
Yes, they frequently coexist, and they synergistically destroy the cow's metabolism.
How does calcium possibly tie into this lipid meltdown?
When a cow gives birth and begins producing colostrum, the first milk, she has to physically pump a massive, staggering amount of elemental calcium out of her blood and into the udder.
Because milk is full of calcium.
Exactly. If her parathyroid gland doesn't respond fast enough to pull calcium from her bones to replace it, her blood calcium levels plummet.
And calcium isn't just structural for bones. It's the chemical key required for muscle contraction, isn't it?
Specifically, it is vital for smooth muscle contraction. When extracellular calcium drops, the smooth muscle tissue lining the entire gastrointestinal tract begins to fail.
Oh, no.
The immense ruminal contractions, the churning of the vat we talked about, simply stop. Intestinal motility drops to 0.
Oh, wow. I see the feedback loop. If the rumen physically stops moving, the cow physically stops eating because her stomach is paralyzed and full, her dry matter intake crashes to absolute 0.
Yes, and a sudden, violent drop in feed intake in a cow that is already in a state of negative energy balance will exponentially accelerate her fat mobilization.
It just pours gas on the fire.
It catapults her from a manageable subclinical state directly into severe, life-threatening clinical ketosis within hours. One metabolic disorder perfectly and ruthlessly triggers the other.
So, as someone listening to this cascade of failures, the obvious question arises. If we know this energy crisis happens every single time a high-yielding cow gives birth, why can't we just preemptively feed them a massive bucket of sugar to stop it? Just artificially plug the charger into the wall.
It seems entirely logical, but it perfectly illustrates how precariously interconnected the ruminant system is. The primary veterinary treatments for a cow already in clinical ketosis do involve glucose precursors.
Like what?
Specifically, oral drenches of propylene glycol, or direct intravenous injections of dextrose, which bypass the stomach to provide the liver with immediate substrate for gluconeogenesis. But you cannot simply overfeed massive amounts of fermentable carbohydrates like corn or starch in her daily diet to prevent the crisis.
Why not? Is it because of the rumen bioreactor's hydrogen limits we talked about earlier?
Yeah. It's because of the pH limits. If you dump massive amounts of rapidly fermentable starch into the rumen, the specific populations of amylolytic bacteria, the starch eaters, will ferment it so quickly and violently that they produce excessive amounts of VFA and, crucially, lactic acid.
And lactic acid is strong.
Lactic acid is much stronger than the VFAs. The pH of the rumen rapidly crashes from a healthy 6.5 down to 5.0 or lower. This condition is known as subacute ruminal acidosis, or SARA.
And acid burns?
Literally. The highly acidic environment physically kills off the delicate cellulolytic bacteria that digest grass. As the bacteria die, they burst, releasing highly toxic lipopolysaccharides, or endotoxins.
Straight into the stomach fluid.
Yes, and simultaneously, the acid chemically burns and damages the stratum corneum of the stomach wall. The tight junctions between the epithelial cells break open, and those deadly endotoxins leak directly out of the stomach and into the bloodstream.
Triggering toxic shock.
Massive systemic shock and lemonitis in the hose.
So treating the liver's energy deficit too aggressively with starch actively crashes and destroys the rumen's microbiome and physical lining. You attempt to save one system and you completely annihilate the other. It really is a biological razor's edge.
It is incredibly fragile.
And the absolute wildest part of all of this, the underlying reason the cow willingly sacrifices her own body fat, damages her liver architecture, and risks systemic immune failure, is purely to synthesize a single specific type of sugar inside her udder.
That's the tragic beauty of it.
Which brings us to the mechanics of milk itself, the synthesis of lactose.
It is the ultimate biological imperative for any mammalian mother, pushed to its absolute physiological extreme in the dairy cow. Milk production isn't a sudden event. It is the final, agonizing phase of a deeply complex developmental sequence.
There are 4 phases, right?
Yes. The mammary gland undergoes 4 distinct chronologically strict phases. The proliferative phase, where cells multiply, secretory differentiation, where they mature, secretory activation right at birth, and finally, the sustained lactation phase.
And the absolute driver of that lactation phase, the single chemical factor that determines the sheer volume of milk sitting in the bulk tank at the end of the day, is lactose.
Correct. Lactose is a disaccharide sugar unique to milk, and it constitutes roughly 5% of bovine milk. But its importance isn't just nutritional. Physically, lactose is the primary osmotic driver of milk volume.
Let's explain that osmotic drive. Lactose is essentially a biological water magnet.
Yes, a very strong one.
The more lactose the cells inside the udder manufacture, the more water is forcefully pulled across the cellular membranes from the cow's bloodstream into the milk ducts to dilute it.
Precisely. The volume of water strictly follows the mass of the lactose to maintain osmotic equilibrium. Let's zoom in on the cellular biology to see how this magnet is built.
Down into the udder.
Deep within the tiny balloon-like alveoli of the udder, inside the mammary epithelial cells, or MECs, there is a structure called the Golgi apparatus. This is where the lactose is synthesized, and the process requires a highly specific transient enzyme complex.
Alpha-lactalbumin.
Yes, alpha-lactalbumin, or LALB. This protein is heavily upregulated immediately after parturition, triggered by lactogenic hormones like prolactin. LALB binds with a pre-existing membrane-bound enzyme called galactosyltransferase.
And they form a team.
Together, they form the functional lactose synthase complex. This molecular machine takes UDP galactose and glucose, strips them of their attachments, and physically bonds them together to form the lactose molecule.
But the limiting factor, the true bottleneck of milk production, isn't a shortage of these enzymes, is it? It's the physical difficulty of getting the raw glucose inside the Golgi apparatus in the first place.
That is the critical cellular bottleneck. We have to look at the enzyme kinetics, specifically the Michaelis-Menten kinetics.
Okay, let's unpack that.
Biologists measure this using a Coulomb value, which represents the substrate concentration required for an enzyme to operate at half of its maximum velocity. It measures the enzyme's affinity or appetite for its substrate.
So a low Coulomb means it's super hungry and grabs the substrate easily.
Exactly. But the GRM of the lactose synthase complex for glucose is surprisingly high. It's around 1.5 millimolar.
Meaning the enzyme has a very low natural affinity for glucose. It won't grab the glucose unless it's absolutely swimming in it.
Exactly. It requires a relatively massive concentration of free glucose inside the Golgi just to operate efficiently. So the absolute fundamental rate-limiting step for making milk is the physical transport of glucose molecules out of the blood, across the basal membrane of the epithelial cell, and across the membrane of the Golgi itself via GLUT transporters.
So we can trace the chain backwards.
Yeah.
If the liver is failing and not making glucose, like we saw in ketosis because the rumen wasn't making propionate, then the blood glucose drops.
Yes.
The Golgi apparatus starves for glucose. The lactose synthase enzyme slows down because of its high collimeter. Less lactose is made, which means a weaker osmotic pull, less water enters the udder, and the total volume of milk physically plummets.
It is a direct, unbreakable biochemical chain connecting the fermentation of grass in the rumen directly to the physical volume of milk.
You know, the sources we reviewed indulged in a really brief cross-species tangent that I think perfectly illustrates how completely lactose dictates the water content of milk. They compared the cow's physiology to a seal.
Oh, it's a beautiful example of comparative evolutionary biology. A cow's milk is roughly 5% lactose. As a direct physical consequence of that osmotic magnet, the milk is about 80% to 85% water.
Very watery.
It is a dilute, high-volume fluid, perfectly designed for a calf that lives in a temperate environment and has continuous round-the-clock access to its mother to stay hydrated.
But a seal pup is born directly onto freezing pack ice.
Right. A seal pup's survival strategy is completely different. It needs to rapidly double or triple its body weight in a matter of days to build a blubber layer so it doesn't freeze to death in the ocean.
Yeah.
It needs massive, hyperdense calories.
And the mother can't give it water.
No. Furthermore, the mother seal is often fasting while nursing. She absolutely cannot afford to lose excessive amounts of water from her own body to hydrate the pup, or she will literally die of dehydration.
So how does she solve the water loss problem? She stops making the water magnet. Seal milk has almost zero lactose.
Exactly. By evolutionarily downregulating the expression of alpha-lactalbumin in her mammary gland, the seal synthesizes almost no lactose. With no lactose, there is no osmotic pull dragging water out of her blood. As a result, seal milk is incredibly thick and dense. It can be up to 50% pure fat with very little water content.
That is just phenomenal. Just by altering the transcription of one enzyme complex, the seal completely alters the biophysical properties of its milk to suit an Arctic environment.
Biology is endlessly adaptable.
So let's trace the incredible journey we've been on. We've seen how the cow uses panoramic dichromatic vision to find food while avoiding predators. We've seen how it ferments that food in a methane-producing, volatile, fatty acid-generating bacterial vat. How it recycles its own toxic nitrogen to feed those bacteria.
An incredibly tight loop.
Right. How it willingly burns its own body fat and risks liver failure to survive the energy deficit. And how it actively channels all of that hard-won glucose into the Golgi apparatus to synthesize the osmotic pull of lactose.
It is a monumental, awe-inspiring orchestration of diverse physiological and biochemical systems.
But what conducts this orchestra? What is the overarching master signal that coordinates all of this? The signal that tells the cow's body when to grow, when to start breeding, and exactly how much milk that udder should be preparing to make.
This is my favorite part.
Prepare yourselves, because this is where the science goes from brilliant to completely counterintuitive. The master signal. is simply the number of hours the sun is in the sky, the master clock, photoperiodism, and neuroendocrinology.
This is perhaps the most profound demonstration of how deeply a mammalian organism is coupled to its planetary environment. Photoperiodism is the physiological reaction of an organism to the length of day or night.
The rotation of the planet.
Yes, and in the bovine, the literal physical rotation of the Earth, translating to the length of the day, dictates lactation, growth, and reproduction.
We're talking about manipulating the light environment. Long day photoperiods, or LDPP, which
Yeah.
equates to roughly 16 to 18 hours of light a day, versus short day photoperiods, SDPP, which is about 8 hours of light. And the data shows that just by changing the light bulbs in a barn to simulate a longer summer day, a dairy farmer can increase a cow's milk yield by 2 to 3 kilograms every single day.
Which is an incredible increase. To understand how invisible photons of light can physically alter gallons of milk output, we have to look deep inside the brain, at the pineal gland.
The pineal gland.
From an evolutionary perspective, the pineal gland is fascinating. It actually evolved from a primitive Cambrian-era photodetector. If you look back 500 million years at certain ancient vertebrates and reptiles, they essentially had a third eye right on the top of their skull to detect shadows and light cycles.
And while that structure sank deep into the center of the mammalian brain over millions of years of evolution, it still acts entirely as a light sensor today.
Exactly. Light photons hit the cow's retina, converting into electrical impulses. This signal travels down a specialized neural pathway called the retinohypothalamic tract directly into the suprachiasmatic nucleus, or the SCN, located in the hypothalamus.
The SCN is basically the clock.
The SCN is the body's master pacemaker. It takes this daylight signal and relays it to the pineal gland, sending a strict command: stop the synthesis of melatonin.
Let's dig into the exact biochemical pathway for making melatonin, because it doesn't just magically appear out of nowhere. It's built from an amino acid.
Yes, melatonin biosynthesis is a multi-step enzymatic process that begins with the essential amino acid tryptophan.
Tryptophan, like in turkey.
The very same. The cell converts tryptophan into serotonin. Then, the critical rate-limiting step occurs. An enzyme with a very long name, R-alkylamine N-acetyltransferase, or NAT for short, takes the serotonin and converts it into N-acetylserotonin. A final enzyme then adds a methyl group to create melatonin.
But ANAT is the master switch in this process. What does light actually do to it?
Light physically destabilizes and destroys the ANAT enzyme. As long as light is hitting the eye, ANAT is degraded, and melatonin cannot be produced.
It just melts away.
The moment darkness falls, ANAT stabilizes, builds up, and the pineal gland starts pumping melatonin into the bloodstream. Therefore, short nights mean a very brief pulse of melatonin. Long winter nights mean a prolonged, sustained, high level of melatonin.
So the duration of that chemical pulse precisely encodes the length of the night.
Exactly. This simple binary chemical code is then broadcast to every cell in the body. Let's trace how this alters milk production first. When a lactating cow is exposed to a long-day photoperiod LDPP, the short, abbreviated melatonin signal causes a massive systemic shift.
And this specific shift boosts milk production by up to 3 kilos a day. How does it do that? My first instinct would be that it increases growth hormone, telling the body to ramp up.
Surprisingly, the data explicitly shows it does not. The galactopoietic or milk-stimulating effect of long summer days is completely independent of growth hormone.
Then what's doing it?
Instead, the reduction in melatonin stimulates the liver to release a massive increase in circulating insulin-like growth factor 1, or IGF-1. This IGF-1 travels to the udder and directly binds to the mammary epithelial cells, powerfully stimulating them to sustain their secretory activity and delaying their natural death rate.
Okay, so long days equal less melatonin, which equals more IGF-1, which equals more milk. That follows a very logical linear pattern. But the literature introduced a massive, glaring biological paradox here. What happens when the cow is in her dry period?
The dry period paradox.
Yeah, this is the roughly 60-day window right before she gives birth, when farmers stop milking her so the udder tissue can involute, rest, and regenerate for the next calf.
This is where the physiology seemingly flips entirely. If you expose a dry cow to a short-day photoperiod, SDPT, meaning you simulate winter with 8 hours of light and 16 hours of dark, she will go on to produce 3 to 4 kilograms more milk per day in her next lactation cycle, compared to a cow that was kept on long summer days during her dry period.
Wait, hold on. Let me process this. Short days mean more darkness. More darkness means a massive pulse of melatonin. And we just established that high melatonin lowers milk production. So why does keeping her in the dark before she calves make her produce massively more milk after she calves?
It is all driven by the mechanics of the prolactin receptor feedback loop. During those short days, the prolonged duration of the melatonin signal actively suppresses the secretion of a different hormone, prolactin, from the anterior pituitary gland.
So circulating prolactin levels drop to near zero.
Okay. The entire body, including the resting udder, is completely starved of prolactin.
Right. And how does biological tissue defensively respond to a chronic deficit of a vital hormone? It massively upregulates its receptor density. The mammary cells, the liver cells, and the immune cells dramatically increase the genetic expression and physical construction of prolactin receptors, PRL, on their cell surfaces.
They are desperately casting a wider net, trying to catch whatever trace amounts of prolactin might be left in the blood.
Oh, I see the trap being set.
When the cow finally reaches the end of her pregnancy and calves, the physical and hormonal shock of parturition naturally triggers a massive, overwhelming tidal wave of prolactin from the pituitary.
And that tidal wave of prolactin crashes directly into an udder that has been artificially hypersensitized with millions of extra receptors.
Precisely. The biological amplifier is turned up to maximum. This amplified signaling cascade causes rapid, massive proliferation of the mammary parenchymal cells. Furthermore, prolactin acts as a vital survival factor.
Meaning it stops cells from dying.
In the absence of prolactin, the body upregulates a destructive protein called IGF-BP5, which induces apoptosis or programs cell death in the mammary cells to shrink the udder. But that massive prolactin signaling upon calving completely suppresses IGF-BP5. It halts the cellular death, ensuring a vastly larger, healthier population of milk-producing cells for the entire rest of the year.
That is entirely counterintuitive and just utterly brilliant. The pineal gland is functioning exactly like a biological router. It takes a raw binary physics signal. Is there a photon hitting the retina, yes or no? And translates it into a complex chemical code that literally dictates whether mammary cells commit suicide or build extra hormone receptors.
It's astonishing.
We can increase a cow's lifetime milk yield simply by leaving the lights on while she's milking, but we absolutely have to turn the lights off when she's resting to mathematically reset the sensitivity of her tissue.
Yes, it perfectly bridges the physics of light with macroscopic agricultural yield. But the photoperiod doesn't just control the udder. It fundamentally controls the survival of the species by regulating the seasonal breeding axis.
Right, because historically, wild bovines needed to perfectly time their pregnancies. Gestation is 9 months. They needed to breed in the late summer or fall, so that the calves were born in the spring, exactly when the grass was green and plentiful to support lactation. How on earth does the melatonin pulse in the brain control fertility in the ovaries?
The biochemical pathway is astonishingly elegant, and it relies on a very specific hijacking of the thyroid system. Melatonin receptors aren't just scattered everywhere. They are highly concentrated in a very distinct, tiny region of the pituitary gland called the pars tuberalis. Melatonin binds here and regulates the release of thyroid-stimulating hormone, or TSH.
Hold on, let me make sure I'm visualizing this correctly. TSH usually travels down the neck to the thyroid gland to control metabolism, but this is a special version.
Exactly. It does not go to the neck. The TSH produced in the pars tuberalis has a completely different molecular glycosylation pattern. It physically cannot enter the general blood circulation.
So where does it go?
Instead, it acts entirely locally within the brain. It diffuses into the surrounding tissue and binds to receptors on highly specialized sentinel-like glial cells called tanycytes, which physically line the base of the hypothalamus.
Okay, so melatonin controls a brain-specific version of TSH, which talks exclusively to these tanycyte sentinels. What do the tanycytes actually do when they get the signal?
During long days when melatonin is low, this specialized TSH signal rises. The TSH binds to the tanycytes and commands them to massively upregulate the transcription of an enzyme called type 2 iodothyronine deiodinase, or simply DIO2.
Deiodinase. Breaking down the word, it means an enzyme that removes an iodine atom.
That is its exact physical function. The tanycytes absorb circulating thyroxine, or T4, from the blood. T4 has 4 iodine atoms and is relatively inactive. The DIO2 enzyme violently strips off one specific iodine atom, converting the T4 into triiodothyronine T3, which is the highly active, potent form of thyroid hormone.
Inside the brain.
This creates a localized, massive spike of active T3 directly inside the mediobasal hypothalamus without affecting the rest of the cow's body.
So it creates a localized hyperthyroid state just inside 1 millimeter of the brain. And what does that localized T3 do?
T3 acts directly on the genome of the surrounding neurons. It regulates the transcription of 2 critical neuropeptides. Kes-S1, which encodes a protein called kispeptin, and RFRP3.
Kispeptin.
Kispeptin is the master, ultimate trigger for mammalian reproduction. When kispeptin is released, it stimulates the neurons to fire a gonadotropin-releasing hormone, GnRH, which travels to the pituitary, releases LH and FSH, and physically kicks off the entire estrous cycle in the ovaries.
So let me trace this entirely to the end. Long days equal low melatonin. Low melatonin allows high brain-specific TSH. High TSH commands the tanny sites to build DiO2. DiO2 strips iodine from T4 to make active T3. The T3 turns on the kispeptin gene, and kispeptin tells the ovaries it's time to release an egg.
You've got it.
The angle of the Earth relative to the sun is directly mechanically translated into the transcription of genes in the cow's hypothalamus.
It proves how deeply, inextricably interconnected the animal's molecular biology is with the rotational mechanics of the planet itself. They are locked together.
So let's step back and look at the whole massive picture we've painted today. We started with the lazy assumption of a simple farm animal chewing its cud. And what we actually found was a creature with a panoramic, dichromatic sensorium designed for constant vigilance, analyzing shadows as physical cliffs and tasting the stress pheromones of its herd.
We found a highly volatile, strictly anaerobic 40-gallon methane-producing bioreactor that balances its pH by burning its own volatile fatty acid fuel. We observed an incredibly thrifty nitrogen economy that recycles toxic urea through specialized transporters just to feed a symbiotic bacterial workforce hugging the stomach wall.
We witnessed an animal that operates on a terrifying metabolic razor's edge, willing to break down its own body fat, risk insulin resistance, and suffer massive liver inflammation, all to ensure it has enough glucose to synthesize the osmotic pull of lactose.
And orchestrating all of this, regulating the scheduled cell death in the mammary gland and the localized thyroid hormone activation in the brain, is the exact literal number of hours the sun happens to be shining on the animal's retina.
The depth of the evolutionary engineering here is truly remarkable. And that leaves me with a final provocative thought to ponder. We have learned today that by understanding these mechanisms, we can manipulate them. By manipulating something as simple as the light switches in a barn to alter the tanny sites in the brain, or by introducing a highly specific lab-grown consortium of propionate bacteria along with a chemical inhibitor like BES into the stomach, we are literally reprogramming the evolutionary biology of the bovine. We can artificially force the tissue to yield more milk, and we can chemically shut down the archaea to halt methane emissions.
We are fundamentally rewriting the thermodynamic and physical outputs of the organism.
Exactly. So, if we can biophysically hack the cow's neuroendocrine master clock and its digestive microbiome to this extreme molecular degree, are we transitioning from traditional animal husbandry into an entirely new scientific era? An era where livestock are treated not just as animals, but as programmable biological platforms? And if so, what are the ultimate biological limits of what we can encode into the physiology of the creatures we depend on? Can a biological system be infinitely optimized, or does the razor's edge eventually snap?
That is the ultimate question that bridges our advancements in molecular biology with the physical reality of the systems we are trying to control.
Thank you for joining us on this incredibly deep dive. True learning is about uncovering the immense, staggering complexity hidden in plain sight. Keep questioning the simple explanations. Keep exploring the mechanics of the world around you. We'll see you next time.